Hello, welcome to MCAT Chat. Today we're going to be chatting about the repair of DNA. So let's look at what are some of the repair options during replication. So DNA polymerases, they're highly involved in DNA replication. They're what catalyze the synthesis of that new strand, and they're highly involved in the entire process. And they themselves can proofread and edit as they work. So they can kind of, just like you would proofread an essay that you've written, they can kind of go over it and read it, and as they're working, delete words that don't make sense or delete nucleotides that aren't supposed to be there and replace them with what should be there. So it's basically just like a proofreading and editing process like you would do yourself. And then the mismatch repair is when it's still during replication, and it's when other enzymes are coming to fix what DNA polymerase didn't detect. So I kind of see this as when you're working, you haven't quite handed in your essay, you're still in the process of creating it, just like you're in the process of replication here with DNA. This is like giving your essay to someone else to edit, and they're just going over and catching what you didn't really pick up on. So this is what these enzymes are doing. That's not a final copy yet, but they're just going over and they're just trying to pick up on what DNA polymerase did not detect and try and fix that. So there is also repair of mutations. This is after replication. So we're not really calling these errors anymore because now they've been finalized. We're going to call them mutations. So this is more along the lines of, we're kind of keeping to this whole essay analogy. It's sort of like when you've handed your essay into your professor and now they're making these changes, or your teacher, whoever. So this is after replication. So you have nucleases, which are going to basically cut and leave a group. So that's my drawing of scissors. I had a really hard time drawing scissors for some reason, but that's the final product. But nucleases are just a group of enzymes that are just going to cut, and they're going to leave a gap. So they're going to cut out sections that are incorrect, and they're just going to leave a space there. And the undamaged strand can act as a template so that DNA polymerases and DNA ligase can come in and work to fill the gap. And if you want to know what DNA polymerase and DNA ligase do, then definitely check out the video on DNA replication. Okay, thanks so much. Bye.